to the physiology of perception in the nervous system that is receiving information. We're going to start with this touch receptor as just an example. The touch receptor is receiving some stimulation from out in the environment, some amount of pressure, and because it is a pressure that I am consciously feeling, that information is definitely going to be sent along the axon of the touch receptor. And you can see, so in this picture, that is labeled as nerve fiber, and the little red arrow showing that there's an electrical signal. And that electrical signal is going to travel to the end of that axon, to the terminal buttons. It's going to get to the synapse, and then we're going to release neurotransmitters, and that postsynaptic neuron in this example, it's going to fire as well, because again, I'm consciously perceiving this, and so that postsynaptic neuron receives enough information that it sends its action potential as well. And we're going to walk through these terms now. I like to do this from a picture. I'm going to keep coming back to this picture. There are several other pictures that I'm going to use to get us through here, but I'm going to make sure I use my words as well, and sometimes just leave the words up. Another thing that you'll notice in this picture is those axons that are leaving each neuron, right? I have a single axon leaving the neuron that's then going to communicate to neurons down the line. In this example, what I've been calling the postsynaptic neuron is receiving information from one axon, from one neuron. This is rarely the way things actually work in the nervous system. A lot of the times in the brain, I as a neuron, I'm going to be receiving signals from probably hundreds of other neurons, or possibly anyway, hundreds of other neurons, maybe thousands. I'm going to send information on. I'm going to have this axon, but it can have these axon collaterals. I can go communicate to maybe hundreds, maybe thousands of neurons. So we're going to keep this really simple with just one neuron communicating to a second neuron. The other thing I meant to really say on that picture with the purple, with the soda, as we were looking at the axon leaving, it had we could see the myelin sheath around it. That myelin sheath, that little bit of purple that we'll come back to, we'll see that same picture again. That's going to allow for the efficiency of communication. It speeds up transmission, and it protects those neurons, those axons, from receiving electrical signals from axons nearby. I've already used some of these words, but I'm going to use them again. So that action potential that we will explain and describe how that's happening and why that's going all the way down the axon, it gets to the terminal buttons. The terminal buttons are at the end of the axon, and they are going to communicate to the neurons down the line. In this case, our single neuron. Usually at the dendrites, sometimes at the soma, and sometimes also at an axon, but we'll leave it at the dendrites, right? So here I get to the dendrites of another neuron. I have my terminal buttons, and I'm going to secrete neurotransmitters. There is this small microscopic gap called the synapse. This is a point where neurons meet or interconnect, but really they're not quite meeting, right? There's this small microscopic gap, and so that change in electrical charge, that can't jump the small microscopic gap, and so they release neurotransmitters, which are chemical messengers that the brain uses to communicate to other neurons. Look at the action potential itself. We're going to look at a single neuron. We're going to do that single cell recording that I introduced in that really super brief history. And I'm first going to start by saying single neurons, they get called nerves. They get called nerve fibers. You may have noticed that in the picture before, but this is different than sometimes when we use the word nerves. We talk about a spinal nerve or a cranial nerve. Those nerves are actually cables of axons. So the optic nerve, the nerve that is taking information from the eye to the brain, it is made up of about 1 million axons from about 1 million cells. And so you just have to keep in mind that this is an ambiguous term. And if I am calling it some kind of cranial nerve, like an optic nerve or an auditory nerve, it's actually made up of many, many nerve fibers or many, many axons. So the optic nerve is carrying signals out of the back of the eye. It consists of axons or nerve fibers of many neurons. And what we're going to do is we're going to record from one of the small nerve fibers within the optic nerve. That's how we're going to look at this action potential. And the way they do this is they'll have a recording electrode that's located where the electrical signal, so where it can actually measure what's happening within that axon. They say it pokes through the axon, but it's actually like just right next to it where I can record and be affected by what's happening within that axon, so where the electrical signals are going to occur. And then we're going to have a second electrode, a reference electrode that's off somewhere in the extracellular fluid where that action potential, that change in electrical signal or electrical potential, it will not affect that particular electrode. And so what we're going to do is we're going to look at the difference in charge between these two electrodes. When we're looking at the action potential and we're saying it's about -70 millivolts, we mean that the inside of that neuron and its resting potential is about -70 millivolts in comparison to the extracellular fluid. So this is a comparison. We're looking at the difference in charge, and we're about to see, I'm going to show a picture with an oscilloscope, which is the next word down there, oscilloscope. It's going to indicate that difference in charge by giving us a small little dot on the screen, and then as that moves across time, it'll create this line. Along the x-axis, we're going to see time moving along. What we're looking at now is if you look at the word meter there, that meter is communicating, we're getting that information to the oscilloscope, which we're seeing over on the right. I'm starting at the top right now. And the meter is going to be giving us or showing us the difference in charge, right, of the recording electrode that is there hitting the axon. So it's going to tell us about the electrical charge within the axon, but it's actually giving us the difference between that recording electrode and the reference electrode out in the extracellular fluid. And so if we look at the oscilloscope when nothing's happening, we're going to call this the resting potential. We're at -70. That's -70 millivolts. And then, so this is the touch receptor, and we're going to feel that touch, and so it's going to send an action potential down the axon. And so that red band, as we magnify that axon, and the red band starts to go by the recording electrode. As it starts to go by the recording electrode, we go up from -70 millivolts and up and up and up to +40 millivolts. That's actually what we call the action potential. And then that recording electrode, which is not moving, right? It's staying at the same exact place. Now, as that action potential continues to go by over time, we're going to go from +40 millivolts and down and down and down back to -70 millivolts. I'm going to share a couple of slides that I stole from Dr. Neil Levins because they were cute and I'm keeping them. So resting, I'm happy when I'm resting. You're happy when you're resting. Resting is good. At rest, we're at -70 millivolts. The other thing this cute picture shows is that it's negative inside in comparison to outside of the axon. The action potential, for whatever reason, appears to be kind of angry, but you can think of it as excited or feeling something or whatever, but I'm awake, action. It's at +40 millivolts, and again, it's positive in comparison to outside. If we look at why this action potential occurs, it's important to start by looking at the components of the neuron's liquid environment. And so what you can see in this picture is there's more sodium. So there's little blue dots that say Na+. Na+ is sodium. There's more sodium outside of the axon than inside. There is more potassium. So the K+, the red dots, are potassium, and it's showing that there's more potassium inside the cell than outside the cell. You can see also they both have a plus after them. They're both positively charged ions. I'm going to ignore the other ions here, but the other ions, basically between the sodium having so much more outside than inside and the other ions that are there, it's negative. It's negatively charged inside the cell compared to outside of the cell. And so if we think about how physics works, those positively charged particles want to get into the cell, right? If we think about that electrostatic pressure, if we take two magnets and they're both positively charged, they'll repel each other. But if one's positive and one's negative, they get pulled together. So basically we're pulling those into the cell because it's negatively charged in there. They're positively charged ions, and so they want to go in. But we also know, right, if I pour a bunch of salt into a glass of water, it's going to diffuse. That force of diffusion means the sodium ions are being pulled in, just like they were by the electrostatic pressure, just like they were by the fact that it's negative inside the cell. Those sodium ions want inside that cell very badly. The potassium ions, though, notice this in the diffusion, they're going to want to go out. There is a slow leakage out of potassium, but we're going to really ignore that. For the most part, what we're going to start to see is right now, with our channels closed, those sodium ions are stuck outside, those potassium ions are kind of stuck inside with a slow leakage out. But what we see with the action potential, it's basically occurring just with physics and the fact that we open up these channels because of the cell's liquid environment. So I'm the touch neuron, I receive we're going to get to this threshold of excitation. I'm going to get enough stimulus energy that, yes, I'm going to send my action potential. And what that means, sending that action potential, what that means is I open these sodium channels. And so sodium flows in. So here's my neuron at resting potential at negative 70 millivolts. I open my sodium channels and sodium flows in. And so now we're going up negative 70 millivolts, up and up and up and up to positive 40 millivolts. And you can see they're trying to show this with the blue part of that band now. That's the sodium flowing in. Sodium channels shut. Potassium channels open fully. I actually did this a little bit earlier, but that's okay. And potassium's flowing out. Notice potassium has a positive charge, and so sodium channels are shut. I'm at positive 40 millivolts, and now the potassium is flowing out, so it's taking its positive charge out of the cell. And so I'm going from positive 40 millivolts and down and down and down back to negative 70 millivolts. We actually overshoot just slightly. So I'd like to do a lot of this from pictures, but it is good to see the wordy words and make sure that I use the terminology, use my language, use my words. So stimulation, in this case, we were talking about a touch receptor, so some kind of pressure or touch stimulation causes the channels in the cell membrane to change their what's called permeability. And permeability is this property of the cell membrane that refers to the ease with which particular molecules can pass through the membrane. So remember, we're first going to change our permeability to sodium. By permeability to sodium, we're going to say that the membrane is selectively permeable to sodium. And again, those positively charged sodium ions are going to flow into the neuron. So we're going to see that shift in electrical charge, right? As those positively charged ions are coming in, we're going to go from negative 70 millivolts up and up and up to positive 40 millivolts. When we get to positive 40 millivolts, the membrane becomes selectively permeable to potassium. I said this in a slightly different way in that I said sodium channels snap shut. Potassium channels, they have been slightly open, but they're going to open fully, and the positively charged potassium ions can now flow out of the neuron. And again, because we're bringing that positive charge now out of the neuron, we are reestablishing the negative charge inside the axon. Here's my picture again. Pause, think. I am going to include a video for the Khan Academy explaining this as well, so you can get another example with some animations going on. If you're like me, one of the things that maybe bothers you at this point is now all the sodium and potassium are in the wrong places for us to be able to have that process occur again. And so this process of the sodium-potassium pump, kicking out some of the sodiums and bringing back in some of the positively charged potassium ions, that process is an active process, and we're just basically putting things back into their original places so that, and their original ratios, so that we can have this process happen again, where we kick out 3 sodium ions, pull in for every 2 potassium ions that we pull in. Again, I want to just reiterate, this is happening after the action potential has done its thing. This is allowing for that action potential to occur again. Some important notes for the action potential. One is that it is a propagated response. Once it's triggered, it fires to the end. It does not decrease in size. It does not change velocity. It is an all or none kind of thing. So this is called the all or none law. If I receive a signal of sufficient strength, and at some point I'm just going to start talking about it as I receive my threshold of excitation. I receive that signal. It triggers this action potential. It starts at the axon hillock, and it goes down all the way to those terminal buttons. If I do not receive a signal of sufficient strength, if the signal is too weak, I just do not fire. That information does not get sent on at all. So it is an all or none law, which means the only way for a neuron to communicate information about the strength of the stimulus, the only way for me to change the strength of the stimulation is to vary the rate at which I fire these action potentials. So I'm going to, if I'm a neuron that goes from negative 70 millivolts to positive 40 millivolts, that's what I do. There's no sometimes going up to positive 60 and sometimes going up to positive 30, depending on strength. If it's a brighter light, I have to just fire faster. I'm going to have that same negative 70 to positive 40 millivolts. I'm going to have that same speed down the axon. If it's a brighter light, I'm going to fire faster. If it's a stronger touch, I'm just going to fire faster. Put this with a picture. So let's just say in A, this is a very light touch. This picture says 1,000 words for me. First, you're going to see that before pressure on occurs, we can see these action potentials. And I'm going to say, so what we saw was negative 70 millivolts up to positive 40 millivolts, and we watched this happen across a line. We're going to squash that, or across time slowly, and it looked like this little almost normal curve. We're going to squash that normal curve into a line, and that line is going to say action potential. So that's the neuron firing. You're going to see a lot of these figures. So in A, I'm getting a very light touch, and you can see before that pressure even comes on, there's this slow, and I'm going to try and do this across time, this slow kind of fire, fire. Pressure comes on. Fire, 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 fire. Okay? Not too quickly, but faster than what I'm going to call spontaneous firing. Neurons do just spontaneously fire. That's what that is showing before the pressure comes on. Our neurons will fire spontaneously. B, we're going to see a medium touch, right? So they're not just barely touching me, but they're actually touching me pretty well. I'm feeling some pressure. I can feel it. Yeah, starting at the beginning of the figure. In B, fire, fire. Pressure comes on. Fire, 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 fire. Okay, a bit faster. C, I'm going to get that touch as hard as I can before causing pain. I used to say it caused pain, but then we're actually in a whole other world of neurons. And so just harder than that, just under pain. In C, a hard touch. Before pressure comes on, fire, fire. Pressure comes on, fire, 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 fire. It's starting to sound like Beavis from Beavis and Butthead, right? Firing as fast as, fire, 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 as fast as you can do it. Okay, so I'm telling you about the strength of that signal, or the strength of the stimulus out in the environment, by firing faster. So we're back to this slide from the beginning that I usually stop and make sure there aren't questions, but I will just point out a few of the things that I have now said with the language on the slides, that we are looking at a touch receptor and we're receiving enough stimulus energy from the environment that we are feeling that touch, and so we reach our threshold of excitation. We're going to come back to that as we talk about receiving information and putting that information together to see, do we reach our threshold? Am I going to send this action potential? And then we talked about the action potential. So if you look at that red arrow and the words electrical signal, that is my action potential. And it's going to go from that axon hillock right next to the soma, the cell body, all the way down the axon to those terminal buttons. And at the synapse, we're going to release neurotransmitters, chemical messengers, to communicate to the postsynaptic neuron, the neuron down the line. The other thing you can see on these axons, that little bit of purplish hue that is surrounding the axon, that is the myelin sheath. You can see the breaks in the myelin sheath where there are these nodes of Ranvier. We don't need to even know those for this class. We're going to ignore that. And so the postsynaptic neuron, since I'm feeling this touch, I'm conscious of feeling this touch, that postsynaptic neuron receives enough information, enough excitatory information, that it reaches its threshold of excitation and sends its action potential, which again we see with the little red arrow, going all the way down the axon to the terminal buttons to communicate to neurons down the line. And I'll say again, this is a single neuron communicating to another single neuron. This is usually much more complicated in the nervous system.